Hello everyone. Today we are going to go through a very quick video on how to run CSPOT. In order to follow through this tutorial, all you need is two files. I hope you have this in hand right now. So here is an example data of all the raw materials that are required to run CSPOT, and also a Jupyter notebook, which we will use to basically go through this tutorial. And very quickly, for running CSPOT, you need three main elements. One is the actual image, the multiplexed image, the segmentation mask, and the quantification or the spatial feature table that was derived from using the image and the segmentation mask. Okay, so before we get started, I just want to take you through the documentation page as well. So all the functions that are within CSPOT are well documented within this section of the page. So you can go through each function, and every parameter would be kind of described here. And if you go towards the end, you'll also have an example of how to run that function. And the easier thing to do is essentially go to the tutorial, from downloading this tutorial data up through running all the different functions and pipelines within CSPOT. So with that out of the way, let me quickly take you through how to run CSPOT. There are multiple different ways of running CSPOT. You can run it either in the command line interface, use Docker, or the way I generally run it, especially if it's like a small data set, is to do it in an interactive manner. So that's what we are going to do now, because this exemplar data set is like really small. But if you're running it in large images, ideally this would be implemented on a high performance computing cluster and run it using the command line interface. We highly recommend using a Conda environment. Let's create a new environment, install CSPOT within it, and then run through the Jupyter notebook. So in order to create an environment, all I'm doing here is opening up a terminal. Again, this is a M2 Mac with the latest version of OS on it. I have tested this on the latest version of Windows as well, and also a normal the Intel chip Mac as well. Create an environment. That's Conda create, give the name of the environment you want to use and the Python version. This is going to take a while that's being created. Once that is created, all we would do is activate that environment like so. There we go. That's created. Let's activate the environment. I'm not sure if this is visible, so let me increase the font size a bit here. So we have activated the environment, and then within the environment, let's install CSPOT, pip install CSPOT. And this is gonna take a while to install it. And once we have installed CSPOT, I'm going to install Jupyter Notebook after this and use Jupyter Notebook to run through the tutorial. So that's it. So CSPOT is installed. Let me now install the notebook. So as you can see, it's pip install notebook. I'm going to do that. Pip install notebook. And that's going to quickly install Jupyter Notebook. Okay, I'm going to download cd into my download folder and initialize Jupyter Notebook. So okay, that's pretty much it. And you can see that Jupyter Notebook is being loaded. I can now just minimize this off. And as you can see, since I cd'd into the downloads folder, it's already within that folder. That's my exemplar data, and here is the Jupyter Notebook that we are going to follow right now. So if we click on that, as you can see, it kind of opens up the notebook. Let me just zoom in a little bit so that it's easier to see. Okay, so again, this also has some information about creating that new environment, people installing CSPOT notebook, and then calling Jupyter Notebook, which we have already done. So let's go further down. So here, the first step is to actually import the package. So that's what we are going to do. In order for you to run CSPOT, you basically need to give in a path to the the folder directory that you're using. An image path, what is the where does the entire image live? So that's essentially this directory here. So we need the project directory, we need the actual image path, and then we would need the segmentation spatial table math, which is nothing but the quantification table here, and the marker channel mapping path, which is the markers.csv. So this is nothing but because CSPOT doesn't know what are the actual layers of channels within your image. So you basically need to tell it that the first layer is DNA, the second layer is ECAD, the third layer is, CD45, and so on. And there's one additional column here, which you don't need it right now, but later on you would need, because the way CSPOT works is you train a model and then you apply that model on a new image. So on a new image, you need to tell CSPOT which marker corresponds to which model. So in this case, we have just named the same. So we are using ECAD for ECAD and CD3 for CD3. Okay, so you can see that that's completed. So we're going to run this. We're just going to initialize all the folder paths. And the first step is to generate exemplar thumbnails. So in this case, we are going to be running for two markers, ECAD and CD3, and then a human goes into that and make sure that, the positive thumbnails are really positive and the negative thumbnails are really negative. Since this is a very quick tutorial just for, making sure that the algorithm works, I will not go into detail. So we have already done a tutorial previously. Maybe I'll upload that on a YouTube channel. Okay, so let's generate the thumbnail. So you can see that it's processing ECAD, processing CD3D. Since we set the project directory as, CSPOT example data, it's going to create another folder within your project directory with all caps CSPOT, and this is going to be the main directory where it kind of saves all the results. So if you go within CSPOT, we can see that now there's a new folder called thumbnail, so which essentially this function has created. And within thumbnails, the kind of two markers that we asked the algorithm to create, ECAD and CD3D. And within ECAD, you can see that there is a true positive folder and a true negative folder. So again, now the objective here is to make sure all the thumbnails within the true positive or really positive. So anything that is ambiguous, just delete them off. And true negative needs to be truly true negative. And again, even if you have the slightest doubt of, if it's negative, just delete them off, because we only need a very small number of good positive and negative examples for the algorithm to learn. I like to keep everything organized, so it automatically creates two additional folders called positive to negative and negative to positive. You'll realize that when you're going through this, true positive, there will be clearly like true negative thumbnails within that. So I generally just move that into positive to negative kind of folder. And also when you're going through the true negative kind of thumbnails, you realize that there are clearly some examples of true positive thumbnail. So I generally take those thumbnails and move it to negative to positive folder. Again, the algorithm kind of knows the folder structure. So when you run the next function, it's going to go through all of these folders and use them appropriately. So I generally do not use the actual raw data. I usually use something called a local norm, which allows me to look at the images a little bit better. And then there is another helper function which will help you to kind of replicate all the actions that you have done here into those main folders, which would eventually go into the model. Again, those are all written in more detail in the actual tutorial, which I'll not go through in detail. The next step is to essentially generate a training and test split for the unit algorithm to start learning on. If you run this, it's going to go through that and create training data for the unit algorithm to train on. So again, now you can see that you have an additional folder here called training data. Let me just go inside this so that it's a little bit easier. So previously we had thumbnail. Now you have another folder called training data. And if we quickly look into that, it's the same thing. Like we have, we have created training data for ECAD and CD3D. And if you go inside CD3D, you can see that there are the general, training, validation, and test split. And if you go into training, so for now, each image would have its own mask. Okay, right. So the next step is to, now that we have created training data, it's to train the actual algorithm. So again, we are going to only do one epoch of training here. So again, this is just for showing how it works. But generally, you would do around, 100 epoch till you get a stability in your training process. Okay, so when we let's run that, and you can see that it's beginning to train now, and it's done because this is a very small data set, and it basically says that go to CSPOT output to see the results. So you can see that, the CSPOT model has been created here. And again, it has created models for both that kind of training data that we supplied it. Okay, so the next step is to predict on a new image and any kind of new experiment which has those models. So we're going to take a new image and then take these models and apply these models on those new image and predict the probability of expression of CD3D or ECAD on those new images. And we are not going to use the model that we just created because this was literally one epoch. It would have not learned anything. We are going to use the manuscript model that I have included within this exemplar data, and essentially this was the model that we trained for the purpose of the manuscript. In the CSPOT model path, ideally you would give this location here. That's the CSPOT model, but in this particular case, as this is a tutorial, I'm giving the path to the manuscript models. And you also need to give in the segmentation path. Okay, so let's initialize those paths, and then let's run the entire CSPOT pipeline. So that's going to be, running a couple of different things. As you can see that it's loading the model, it's doing some inference, it's going to save those models as a probability mask. It's going to take that probability mask, apply the segmentation mask on it, and identify, the median probability for every single cell based on those segmentation masks. And then it's going to combine those probability scores with the actual, quantification spatial table scores, and then run a series of kind of machine learning algorithms, and then do a final prediction of which cells are positive for ECAD and which cells are positive for, in this case, CD3D. So, okay, so you can see that it has created a lot more folders here. So currently it kind of saves every intermediate step. But in the future, we might remove this to kind of have only the last, final output. But in this case, what you're essentially looking for, the final kind of output is saved within CSPOT output. So that's where I would go. And you can see that there's one file there, which is in the format of H5AD, which is an AND data object. Essentially, it would contain the predictions that were made. So in order to visualize the predictions, we are going to just do a very simple scatter plot here. And this is kind of the predictions that were made. Those are the ECAD positive cells, CD8 and CD45. And that kind of, ends CSPOT. It would have also saved a normalized data matrix for all the markers that you have run CSPOT on. So you can do any kind of downstream analysis that requires a normalized data matrix. So just for the sake of completeness, we also include a phenotyping kind of approach here. And this works in a more prior knowledge. So for example, in this case, you can see that I'm going to take all the cells and then divide them into immune and ECAD positive cells. ECAD positive cells are marked by expression of ECAD, while immune cells are marked by the expression of either CD45, CD4, CD3, or CD8, right? And then I can take the ECAD positive cells and then subclassify them further. In this case, I'm going to just classify them as Ki67 positive ECAD positive cells. In this case, since they are already ECAD positive, I don't have to, reiterate that. I just have to put positive for Ki67. Similarly, I'm going to take immune and then subclassify them further into CD4 T cells, CD8 T cells, and so on. Again, these are all, explained in more detail in the documentation and tutorial page. So feel free to go through those documentation to get an idea of how to use this part of the algorithm. So all I'm doing here is I'm loading that phenotype file, which I created in Excel, and then I'm going to run the actual phenotyping algorithm. So you can see that it's phenotyping the immune cells, ECAD, subsetting the ECAD cells, and phenotyping ECAD Ki67 positive cells. It's subsetting the immune cells and then further classifying them into different categories. And once that is done, it's going to save final identified phenotypes into a separate column within the AND data object called phenotype. And this is just for illustration purpose, but that's how one would go about, doing phenotyping. So that's pretty much it. That's how you run CSPOT. If you have any questions, feel free to leave a comment in the GitHub page, and I'll be able to help you out and answer questions there. but you should be able to find a lot of examples and detailed tutorials. I did spend a lot of time trying to write this out, so I will highly recommend reading them carefully to get a sense of how the algorithm works and how to kind of manipulate each parameter. And I hope to also upload a more detailed tutorial video going through the process of looking through those thumbnails and how do I gently sort them out into positive thumbnails and negative thumbnails. Gently just open two different folders here, one with positive and the other with that positive neg, and I just drag around positive to neg folder and delete off any ambiguous examples there. So that's how I do it. So if you have any questions, feel free to ask me. Thank you for listening. If you enjoyed the tool, thank you.